So far, our study of the endocrine system has taught us the basic structure, classifications, and effects of hormones, as well as the feedback loops and specific hormones released by the hypothalamus-pituitary complex. Today, we will be examining hormones released from other glands and tissues in the body. We will begin with the largest pure endocrine gland in the entire body, the thyroid gland. The thyroid gland is located in the anterior neck, on the trachea, just inferior to the larynx. Its butterfly shape allows it to straddle the trachea and sit close to the major arteries on each side of it. A median mass called the isthmus connects the two lateral lobes. Internally, the gland is composed of hollow, spherical follicles, shown on the right. The walls of each follicle are mostly composed of cuboidal or squamous epithelial cells called follicular cells, which produce a glycoprotein called thyroglobulin. The inside of these hollow cavities stores colloid. This is an amber-colored sticky material consisting of thyroglobulin molecules bound to iodine atoms. The hormone produced by the thyroid gland, cleverly named thyroid hormone, is made from this iodinated thyroglobulin. The parafollicular cells, which can be seen in the circle outside of the follicles, produce calcitonin, which we will not discuss in this lecture, but that you'll need to read about on page 615 of your textbook. Thyroid hormone, or TH, produced from the iodinated thyroglobulin, is often referred to as the body's major metabolic hormone. It is actually two different iodine-containing amine hormones called thyroxine, or T4 because it has 4 attached iodine atoms, or triiodothyronine, or T3 because it has 3 attached iodine atoms. T4 is the major hormone secreted by the thyroid follicular cells, while T3 is formed at most of the target tissues by converting T4 to T3 and removing the extra iodine atom. TH affects pretty much every cell in the body. Even though it is an amino acid-based hormone, it is able to enter a target cell, bind to intracellular receptors within the cell's nucleus, and initiate transcription of mRNA for protein synthesis. The main functions of TH include increasing basal metabolic rate and body heat production by turning on the transcription of genes concerned with the breakdown of glucose. This is called the hormone's caloriogenic effect. The second main function is regulating tissue growth and development. Thyroid hormone is critical for normal skeletal and nervous system development and maturation. It is also critical for reproductive capabilities. The third and final main function of thyroid hormone is maintaining blood pressure by increasing the number of adrenergic receptors in the blood vessels. While these three are the major effects, this table found on page 612 of your textbook lists more specific effects. The thyroid gland is the only gland in the body that is able to store its hormone extracellularly and in large quantities. A normal, healthy thyroid gland stores enough colloid to provide normal levels of the hormone for 2 to 3 months without replenishment. When thyroid-stimulating hormone, TSH, from the anterior pituitary binds to receptors on follicular cells, their first response is to secrete stored thyroid hormone. Their second response is to begin making more colloid to refill their depleted stock. As a general rule, TSH levels are lower during the day, peak just before sleep, and remain high during the night. As a result, thyroid hormone release and creation follows a similar pattern. The diagram here walks through the second response of the follicular cells, the creation of more colloid. Let's walk through it together. First, after being synthesized on the ribosomes of the thyroid's follicular cells, thyroglobulin is transported to the Golgi apparatus, where certain sugar molecules get attached, and the thyroglobulin is packaged into transport vesicles, shown here in green. These vesicles move to the apex of the follicular cell, where they discharge their contents into the follicle lumen to become part of the stored colloid. In order to produce the functional iodinated hormones, the follicular cells must accumulate iodides, or anions of iodine from the blood. This trapping depends on a form of active transport. Once trapped in the follicular cell, iodide then moves into the lumen by facilitated diffusion. At the border of the follicular cell and the colloid, the iodides are oxidized by the removal of electrons and converted to iodine. The newly converted iodine is attached to tyrosine amino acids that form part of the thyroglobulin colloid. This iodination reaction occurs at the junction of the follicular cell and the colloid. Enzymes in the colloid, like monoiodotyrosine, link monoiodotyrosine and diiodotyrosine together. Two DITs result in T4, while one MIT and one DIT produces T3. At this point, the hormones are still a part of the thyroglobulin colloid. To secrete the hormones, the follicular cells must reclaim the iodinated thyroglobulin by endocytosis and combine the vesicles with lysosomes. Lysosomal enzymes cleave T3 and T4 from thyroglobulin, and the hormones are able to diffuse from the follicular cell into the bloodstream. Again, the main product secreted here is T4, while most T3 is created at the target cells. Now that we've covered the thyroid gland, it's time to move on to the parathyroid glands. These tiny yellow-brown parathyroid glands can be seen on the posterior part of the thyroid gland. There are usually 4 of these glands, but that number can and does vary slightly from person to person. The parathyroid's glandular cells are arranged in thick branching cords containing scattered oxyphil cells and large numbers of smaller parathyroid cells. It is these smaller cells that actually secrete parathyroid hormone. The function of the oxyphil cells is yet unclear. Parathyroid hormone, or PTH, is the single most important hormone in controlling calcium balance in the blood. Precise control of calcium levels is critical because calcium homeostasis is essential for so many bodily functions, including the transmission of nerve impulses, muscle contraction, and blood clotting. Falling calcium levels trigger PTH release, and rising blood calcium levels inhibits its release. PTH increases calcium levels in the blood by stimulating 3 target organs: the skeleton, the kidneys, and the intestine. The flowchart here walks you through each of the reactions. As you can see, PTH release stimulates osteoclasts, or bone resorbing cells, to digest some of the calcium-rich bony matrix and release ionic calcium and phosphates to the blood. It also enhances reabsorption of calcium in the kidney tubules. And finally, it promotes the activation of vitamin D, thereby increasing absorption of calcium by the intestinal mucosal cells. Vitamin D is required for the absorption of calcium from food, but first the kidneys must convert it to its active vitamin D3 form. PTH stimulates this transformation. Now we will move on to the adrenal glands. The adrenal glands are a pair that sit on top of the kidneys, where they are in a fibrous capsule and a cushion of fat. Each adrenal gland is structurally and functionally 2 separate endocrine glands. The inner adrenal medulla, which is more like a knot of nervous tissue than a gland, is actually part of the sympathetic nervous system. The outer adrenal cortex, around the medulla and forming the bulk of the gland, is glandular tissue that is developed from the mesoderm. Each region produces its own set of hormones, but all hormones released here help humans cope with stressful situations. For our examination of this gland, we will begin in the outer layers and work our way inward toward the adrenal medulla. The adrenal cortex synthesizes over 24 steroid hormones, collectively referred to as corticosteroids. The synthesis pathway of these begins with cholesterol and involves varying intermediates depending on which hormone is being made. Unlike amino acid hormones, steroid hormones are not stored in cells. Therefore, they can only be released as quickly as they are made. The large cortical cells are arranged in 3 layers. From the outside in, these are the zona glomerulosa, which produces mineralocorticoids. These are hormones that help control the balance of minerals and water in the blood. The zona fasciculata is next. This layer's cells are arranged in linear cords and mainly produce metabolic hormones called glucocorticoids. And finally, we have the zona reticularis. These cells are arranged in a net-like pattern. They mainly produce small amounts of adrenal sex hormones called gonadotropins. Let's take a look at each of these classes of corticoids before we move on to the adrenal medulla. Mineralocorticoids regulate the electrolyte or mineral salt concentration in extracellular fluids. Particularly of interest are sodium and potassium. Recall from our study of the nervous system that the single most abundant cation in the extracellular fluid is sodium, and the amount of sodium in the body has a large role in determining the volume of extracellular fluid, because where sodium goes, water goes. The regulation of both sodium and potassium is the primary job of a hormone called aldosterone, which reduces the excretion of sodium from the body. It does this by stimulating sodium reabsorption, which in turn increases blood volume and blood pressure, and by causing potassium to be excreted into the tubules for elimination from the body. Aldosterone accounts for 95% of all mineralocorticoids produced and is absolutely essential to life. Its effects are relatively short-lived, allowing plasma electrolyte balance to be precisely controlled and constantly modified. One of the most important mechanisms in the body is pictured in the diagram here. It's called the renin-angiotensin-aldosterone mechanism. It influences both blood volume and blood pressure by regulating the release of aldosterone and therefore sodium and water reabsorption by the kidneys. Please follow along in this diagram and submit any questions you have to the discussions thread for the endocrine system on Canvas. Also, read through and make sure you understand the significance of ACTH and ANP in aldosterone production. These can be found on pages 618 and 619 of your textbook. Glucocorticoids are the next class. These influence the energy metabolism of most body cells and help us resist stressors. Under normal circumstances, they help the body adapt to intermittent food intake by keeping blood glucose levels fairly constant, as well as help maintain blood pressure. This class includes hormones such as cortisol, cortisone, and corticosteroid, but only cortisol is secreted in significant amounts in humans. As with all steroid hormones, glucocorticoids act on target cells by modifying gene activity. Negative feedback regulates glucocorticoid secretion. Cortisol release is promoted by ACTH. ACTH release is triggered, remember, by the hypothalamic releasing hormone CRH. Rising cortisone levels feed back to act on both the hypothalamus and the anterior pituitary, preventing CRH release and shutting off ACTH and cortisol secretion. Cortisol secretory bursts are driven by patterns of eating and activity. It also occurs in a definite pattern throughout the day and is often studied as part of circadian rhythms, the sleep-wake cycle that your body goes through on a daily basis. Acute stress interrupts the normal cortisol rhythm. Higher CNS centers override the inhibitory effects of elevated cortisol levels and trigger further CRH release. The resulting increase in ACTH blood levels causes an outpouring of cortisol from the adrenal cortex. To give you a real-world example of this, let me tell you a short story about an experiment performed in one of my labs in college. About 5 weeks into the semester, we began collecting saliva samples at the beginning, middle, and the end of every day, while recording the time, our mood, and our stress levels at the time of collection. We collected the samples for 2 weeks, just enough time to get us through the first round of college exams. As stress levels rose because we worried about exams, so did our cortisol levels. In fact, some of us exhibited such drastic swings in our cortisol levels that we could not distinguish the typical daily pattern of high in the morning and low in the evening. Typically, by the evening, we were so stressed out that our cortisol levels were even higher than they were in the morning. Make sure you read through the actions of the glucocorticoids to understand how they regulate metabolism. This information can be found on pages 619 to 620 of your textbook. You'll also want to read through the information on gonadocorticoids on page 620. Now let's move into the innermost portion of the adrenal gland, the adrenal medulla. The spherical medullary chromaffin cells crowd around blood-filled capillaries and sinusoids and are modified postganglionic sympathetic neurons. These cells are responsible for making the catecholamines, epinephrine and norepinephrine, that we've already studied as neurotransmitters. When a short-term stressor activates the body to fight or flight status, the sympathetic nervous system is mobilized. Blood vessels constrict and the heart beats faster, together raising the blood pressure, and blood is diverted from temporarily non-essential organs to the heart and skeletal muscles. Blood glucose levels rise, and preganglionic sympathetic nerve endings weaving through the adrenal medulla signal the release of catecholamines, which reinforce and prolong the fight-or-flight response. Unequal amounts of the two hormones are stored and released. Approximately 80% is epinephrine and 20% is norepinephrine. With few exceptions, the two hormones exhibit the same effects. To see these exceptions, read page 621. The final endocrine gland that we will actually study is the pineal gland. This tiny pine cone-shaped gland hangs from the roof of the third ventricle in the diencephalon. Its secretory cells, called pinealocytes, are arranged in compact cords and clusters. Although many peptides and amines have been isolated from this tiny gland, its only major secretory product is melatonin. This is an amine hormone that is derived from the neurotransmitter serotonin. Melatonin concentrations in the blood rise and fall on a daily cycle. Peak levels occur during the night and make us drowsy, while the lowest levels occur around noon. The pineal gland indirectly receives input from visual pathways concerning the intensity and the duration of daylight. In some animals, mating behavior and gonadal size vary with relative lengths of light and dark periods, and melatonin is responsible for these effects. Now that we've examined the endocrine role of the hypothalamus and of glands dedicated solely to endocrine functions, we now need to consider a set of organs that contain endocrine tissue, but that have other major functions. These include the pancreas, gonads, and placenta. We'll begin with the pancreas. This organ is located partially behind the stomach. It's a soft tadpole-shaped organ that is a mixed gland composed of both endocrine and exocrine gland cells. Along with the thyroid and parathyroids, it develops from an outpocketing of the epithelial lining of the gastrointestinal tract. Acinar cells, forming the bulk of the gland, produce an enzyme-rich juice that is carried by ducts to the small intestine during digestion. Among the acinar cells are approximately 1 million pancreatic islets, or tiny clusters of cells that produce the pancreatic hormones. The islets contain two major populations of hormone-producing cells. the glucagon-synthesizing alpha cells, and the more numerous insulin-synthesizing beta cells. These cells act as tiny fuel sensors, secreting glucagon and insulin appropriately during the fasting and fed states. Glucagon, a 29 amino acid polypeptide, is an extremely potent hyperglycemic agent. One molecule of glucagon can cause the release of 100 million glucose molecules into the blood. The major target of glucagon is the liver, where it promotes the following three actions. First, it can break down glycogen into glucose. Second, it can synthesize glucose from lactic acid and from non-carbohydrate molecules, a process called gluconeogenesis. And finally, it can release glucose to the blood from the liver cells, causing blood glucose levels to rise. The secondary effect is to lower blood levels of amino acids, as the liver sequesters these molecules to make new glucose molecules. Insulin, a 51 amino acid polypeptide, is used to lower blood glucose levels, but it also promotes protein synthesis and fat storage. Circulating insulin lowers blood glucose in three ways. First, it enhances the membrane transport of glucose and other simple sugars into most body cells, especially into muscle and fat cells. Second, it inhibits the breakdown of glycogen into glucose. And third, it inhibits the conversion of amino acids or fats to glucose. These inhibiting effects counter any metabolic activity that would increase the plasma levels of glucose. Now, insulin is not needed for glucose entry into the liver, kidney, and brain tissue, all of which have easy access to blood glucose regardless of the insulin levels. However, it does have several important roles in the brain. It participates in neuronal development, feeding behavior, and learning and memory. Insulin secretion is stimulated by four factors. Elevated blood glucose levels, this is the chief controlling factor. Rising blood levels of amino acids and fatty acids. Acetylcholine released by parasympathetic nerve fibers. And hyperglycemic hormones such as glucagon, epinephrine, growth hormone, thyroxine, or glucocorticoids. This effect is technically indirect and occurs because all of these hormones increase blood glucose levels. The next organs we need to look at that contain endocrine tissue are the gonads and the placenta. The male and female gonads produce steroid sex hormones, identical to those produced by the adrenal cortical cells. The major distinction is the source and relative amounts produced. As described earlier, gonadotropins regulate the release of gonadal hormones. The paired ovaries are small oval organs located in the female's abdominopelvic cavity. Besides producing ova, or eggs, the ovaries produce several hormones, most importantly estrogens and progesterone. Estrogens are responsible for maturation of the reproductive organs and the appearance of secondary sex characteristics of females at puberty. Acting with progesterone, estrogens promote breast development and cyclic changes in uterine mucosa, or the menstrual cycle. In male testes, located in an extra-abdominal skin pouch called the scrotum, produce sperm and male sex hormones, primarily testosterone. During puberty, testosterone initiates the maturation of male reproductive organs and the appearance of secondary sex characteristics and sex drive. In addition, testosterone is necessary for normal sperm production and maintains the reproductive organs in their mature functional state in adult males. The placenta is a temporary endocrine organ. Besides sustaining the fetus during pregnancy, it secretes several steroid and protein hormones that influence the course of the pregnancy. Placental hormones include estrogen, progesterone, and human chorionic gonadotropin, or HCG. This table can be found on page 626. You should study it and the material covered on it on pages 625 and 625 through 627 of your textbook. This information gives you examples and feedback loops of hormones produced by organs other than the major endocrine glands, the pancreas, the gonads, and the placenta. This information can and will be on any future tests.